So here's a demo for the disinfectants lab. For this lab, we are evaluating the impact of various disinfectants on the growth of different types of organisms. We usually evaluate gram-positive bacteria, Micrococcus luteus, Bacillus subtilis or Bacillus cereus, and also gram-negative bacteria, E. coli, and others, perhaps Serratia. In this exercise, we will start with a confluent lawn of bacteria, as you've already seen. For this one, we will be labeling the back of the TSA plate with each of the disinfectants that we will be using. So we have 6 different disinfectants. You'll see the list in the Canvas module. And you can see them here, so I have them in order. I'm not going to give you a demo of all of them, but I'll show you how this would be done. Here's bleach, hydrogen peroxide, a 70% ethanol, Rocol D, which is the same disinfectant used in the laboratory to disinfect the lab benches, Formula 409, and Lysol. So each of these works a little bit differently, as you would read in Chapter 9 on microbial control methods. You'll also see I have an egg dropper plate. This is a sterile plate that I'll use to get these disinfectants onto a disc. So here is a set of sterile discs that we have that we'll be using to saturate with disinfectant and put those things on the TSA plate. And then the last ingredient you'll see here, this is a bottle or a beaker with 70% ethanol in and some forceps. Now ethanol is a sterilant, so right now those forceps are sterile. I could use them, but that alcohol can also kill the bacteria or can cross-contaminate. So I have my open Bunsen burner flame in our chemical fume hood so that I can just essentially eliminate off that ethanol from these forceps. So to do this exercise, I start with my forceps. I'll simply pass them into the flame to eliminate out or burn off the rest of that ethanol here. With this, I will take a sterile disc. And these discs are just very small, essentially pieces of filter paper. I'll put this disc in my first dropper dish here, and I'll add my first disinfectant, right? So for example, I'm adding bleach to this one. I'll simply put a drop of bleach into this to saturate the disc. With the saturated disc, and to make sure that there is no dripping from this disc, I'll find the same section on my TSA plate, and I'll simply add the disc to the TSA plate right in the center of that pie-shaped wedge. With this, I'll just press it down, and the disc is now on my TSA plate. From here, I'll put my forceps back in the ethanol to sterilize them, and I'm ready to move on to my next one. I'll give you just one more demo of the same practice. So again, with my forceps, I'll pass them through that Bunsen burner flame and allow that ethanol to burn off. Take my sterile disc, set this one in a new well. Again, add my disinfectant. And with my forceps, I'll take my disc, again making sure that it's not dripping, and put it directly onto the agar. And I'll just press it down lightly, and now I have my disc on the agar. This exercise you will continue until you've moved through all of these different types of disinfectants and saturated all of the discs to put them on the agar. As soon as this is done, this will stay inverted, taped around the edges, and put into 37-degree incubator for a 24-hour period. And then we will be evaluating the zone or the circle around each of these discs that we call the zone of inhibition, and we'll give you a couple of demonstrations or a couple of plates to show you what these zones of inhibition look like. And so in this exercise, we're just evaluating a relative comparison of the zones of inhibition with a circular clearing around each of the discs for all of these disinfectants, kind of relating one to the other with each bacteria, and then from one bacteria to another to see if there is an impact from gram-positives to gram-negatives, for example.